of macrophages affects their function and, again, their environment. So I think in the last 2 years alone, there must have been 15 papers just in Science, Nature, and Cell just on metabolic changes in macrophages and how that defines lineage and function and disease and whatnot. So an interesting topic. So what are we interested in? So my lab is really interested in understanding how metabolic disorders interact with monocytes. Because if you think about it, that is where everything you eat, everything you do interacts with your innate immune system. Okay, and this question actually arose from the very simple question, and something we study in the lab, but I won't talk about today. We actually had my graduate student talk about it yesterday. Why are healthy diets healthy? Even though a lot of these compounds that are supposedly healthy never make it into a tissue. They'll show up in the blood, but they'll never make it into a tissue. They're cleared way too fast, and yet you have health benefits. So this is where that question came from. And I guess one of the more interesting questions, simple questions, but one I think that still hasn't been properly answered, is how do acute inflammatory processes turn chronic? Okay. There are a lot of ideas out there, and as you'll see as it turns out, I think there are some commonalities, and one of the commonalities are going to be monocytes and macrophages. And I'll give you an example at the end. This is where we come to question number 3. Is there a common link between obesity and cardiovascular disease? It sounds like a really stupid question, but it's actually not. And we'll see that there probably are some unexpected links to this. All right, so I quickly want to introduce you to a paradigm that I already introduced for those of you who've been here 3 years ago when I was here the last time. Monocyte priming. I stole this word actually from the microbiologist. Monocyte priming with endotoxin means nothing else that the cell is exposed to a stimulus and remembers that stimulus, and when a second or the same stimulus comes back, they react different than the first time. Okay, so it's a memory effect. And I'll show you that monocytes have memory for metabolic environments, and that has some very interesting consequences, probably mostly detrimental consequences for the animals and probably the humans. I'm going to talk a little bit about glutathione and redox signaling, because for those of you who are not familiar with glutathione and don't know what protein glutathiolation is, I'll give you the 10 most important words and concepts so we can follow through. As Nick said, we've done a lot of work on protein glutathiolation and how it affects proteins, and sort of validation of principle that this is indeed a way to regulate proteins. It's actually a signaling paradigm. And I'll just go through one related to an interesting phosphatase that it turns out is probably a very important central regulator of monocytes and macrophages, but it's also, in a way, or it appears to be, a sensor in monocytes of metabolic stress. And it sort of seems to be a very early modification of this phosphatase, as you'll see, is a very, very early step in what we tend to refer to as metabolic disorders. And I would put to you that the change in this enzyme and these early events really set the stage for what happens days, weeks, months, and maybe years later. Okay? We'll go to Thiol oxidation. I'll explain to you a little bit what we consider redox signaling, and we came across a very interesting phenomenon. Does anybody believe me if I would tell you that thiol oxidative stress is a sexually dimorphic? So you can actually take macrophages from male and female mice, and you get different responses to the same oxidant. Strange, right? No hormones involved, no nothing, but it is the case, so I'll show you that. We don't quite know how it works, but I just want to show you some data. This is all unpublished, including most of this. And then finally, I want to talk to you about glutarredoxin 1, an enzyme that's been around for a long time, looked at for a long time. I bet these mice have been in the bar shop for ages, but they never showed a real phenotype. And I'll tell you why they didn't show a phenotype, because nobody waited long enough. But my graduate student was good enough to take a close look and made an interesting observation and made a very interesting story, and hopefully we'll get to that at the end, and I'll tell you about that. So, for those of you who do not like monocytes and macrophages, you're going to have to sit through 2 slides, and I'll give you a little bit of an introduction. So, this is actually a slide I took from Simon Gordon, one of the experts, true experts in macrophage biology. He's been around for a very long time. This is from 2005, and it's already outdated. Okay. The Mononuclear-Phagocyte System, as it's called, has really 3 sets of cells and origins of cells. As you all know, the bone marrow is where monocytes come from. They enter the bloodstream, circulate, and until a couple of years ago, I thought they also have the same half-life, about 2 weeks. Actually, that's not true either. We know that's not true. Inflammatory monocytes actually have a half-life of about 1 to 2 days before they enter into the spleen, and then they can be released from there again. But this is what you all know as traditional way, differentiation pathway of a monocyte, which then can differentiate into a macrophage. We have different flavors of monocytes. Whether this is true or not is not quite clear. We have inflammatory monocytes that are supposedly the ones recruited into sites of inflammation. And then we have the resident monocytes, or better term would be patrolling monocytes. Those are the ones that actually crawl along the endothelium. And they can extravasate into the tissue, but they can also come back out. There are some fabulous movies on this, how they can enter into the endothelium, crawl at the basolateral side of it, and go back into the bloodstream. They're probably doing monitoring functions, looking for infections and parasites and whatnot. What most people probably don't know is that all your key resident macrophages do not come from the bone marrow. They're actually originating from the yolk sac. So they get developed very, very early on. They make up most of the resident macrophages. Again, in 2005, they were still classified as primitive macrophages, but for example, Kupffer cells, Langerhans cells, peritoneal macrophages, a whole set I'll show you actually here. I think I have a list. Many of those, if not all of them, are actually resident cell macrophages, the truly resident ones. They originate in the yolk sac. They do not come from the bone marrow. Okay? So we have 2 fundamentally different types of macrophages. We have the ones that are monocyte-derived, and we have those that are resident. Okay? And anyway, if you have any questions, we can address that a little later. So let me go straight here. So the process my lab is interested in is really the fundamental first step in any kind of inflammatory process that is driven by monocytes. And that is the detection of a blood monocyte of a chemoattractant gradient and or a injured endothelium. And what they do is the combination of adhesion to adhesion molecules expressed on injured endothelium plus the chemoattractant gradient recruits this cell into the vessel wall. Okay, these can be small vessels, big vessels, but fundamentally this is a process that happens everywhere where you have an injury, tissue injury. Okay, and here is the key chemoattractant, in this case MCP1. We just picked this one because that's your primary chemoattractant involved in the early phases of atherogenesis and the development of atherosclerotic lesions. CCR2 is the receptor. By the way, if you knock out CCR2, or, more importantly, if you eliminate all blood monocytes, at least mice do not develop even the slightest atherosclerotic lesions. So monocytes are a prerequisite for these kinds of diseases, okay? They drive everything. As much as I know a lot of you love T cells, sorry, they're just secondary players. The monocyte may be I always say macrophages do a lot of things. They do nothing well. So they're a little bit stupid, but they're designed that way, okay? But they know a lot of things, a little bit of everything. And so their role is really to detect and report, and then the specialists come in, i.e. the T cells and all the other leukocytes. The smart cell, that's right. Okay, so because this is such a fundamental pathway, this is what we use to look at monocyte function, okay? And I'll get back to that a couple of times. And so if you go and look in papers of people with metabolic disorders or diabetes, you can see it's well known, been known for 100 years, that monocytes are different in these people than healthy people. They have a different cytokine profile, they look different, they actually make more hydrogen peroxide. Neutrophils and diabetics actually make less, so they're a little unique. They lose phagocytic activity. There are more macrophages in wounds of diabetics, yet they don't heal. We know that monocyte counts go up. If you make a mouse or a patient dyslipidemic, they develop monocytosis, and the ratio of these not arbitrarily, but these selected subpopulation of monocytes, in the mouse they're called Ly6C high and Ly6C low, or pro-inflammatory and patrolling. In humans, they're CD14 positive or CD14 low, CD16 positive. This concept is found in many, many mammals. That ratio goes up. Okay, so a lot of things change if you have metabolic disorder. That is not new and in itself interesting, but it wasn't as important as the finding that what changes is the chemotactic activity of monocytes in diabetics. And that's interesting because it's actually a gain of function. And a lot of other things, you have a loss of function. They become dysfunctional, don't do things. And the reason it's important, and that's very easy to understand, is that the number of monocytes you want at the site of injury probably should be highly controlled. Okay, and this is actually a very nice set of experiments, or if you will, an extrapolation of experiments from the group in Boston, where they look at the quality of healing of a myocardial infarction. Okay, and they randomly set this green star here as normal healing in a wild-type mouse, or let's say a healthy patient, although healthy patients don't get MIs. And if you go and you reduce the number, so here's the number of inflammatory monocytes, and here's the quality of healing. And if you reduce monocytes, okay, either by depleting them, okay, or by removing the spleen, and what that does is remove these Ly6C high monocytes, or if you put patients on steroids, okay, you have a reduction in monocytes, and in general, that impairs healing. On the other side of the equation, if you have too many monocytes, that isn't good either, okay? So if you have patients with high levels of these inflammatory monocytes, or patients, again, with monocytosis in general, that's been long known. If you develop monocytosis after an MI, that was always a bad sign. 100 years ago, they already reported that. And in this case, these are ApoE, so this is an atherosclerosis model, a murine atherosclerosis model. If you develop monocytosis in them, the MI actually will heal slower as well. So clearly, controlling the number of monocytes is important. And so increasing their responsiveness to a chemokine is very likely not a good idea, because how is the tissue going to know that it's going to have to cut down on the number coming in, right? And so based on this, we developed the... yes,
how do you define healing?
In the article, that's a good question. I'd have to go look how exactly this is out of...
Is it rate of healing of whatever, or...
No, I think it's, they said quality. So it's not just the speed, it's also the degree of regaining of function. In the mouse, at least. So what I'm going to propose to you is this following paradigm, okay? So this is actually a very beautiful picture from a friend of mine, Michael Rosenfeld, and this is a blood monocyte, I believe in a rabbit, that is starting to extravasate and squeeze in between two endothelial cells. Okay, and by the way, the electric resistance across this endothelial layer will never be interrupted. These cells can squeeze through without any leakage of liquid. They just force their way in, and it has to be that way because if blood would leak in, you get a blood clot, right? So this is actually amazing considering they even sort of drag their nucleus in with them. So here's what I'm going to propose to you. So in a vessel, any vessel of your choice, and you have an injury, let's assume it's a large vessel, chances are the large vessels are going to make MCP1 in response to those injuries. One of many inflammatory mediators that are being released. And we know that monocytes respond to that and get recruited, and then the healing process can start. Healing process starts in general with an inflammatory phase that then translates into an inflammation resolving phase, and then after everything is done and over with, you should have healed and repaired tissue. So what happens under conditions of metabolic stress or disorders? Well, we know that itself actually increases the inflammation associated with this injury, okay? And so it's no surprise that you actually see more MCP1 being made. And so it's also no surprise that would cause more monocytes to be recruited, okay? So far, nothing special. That wouldn't be very interesting. But what we're proposing is that there is a second component to this. And the second component is that the metabolic stress alone is sufficient to already transform these healthy monocytes into dysfunctional monocytes, or primed monocytes, okay? And what I'll tell you and show you is that these primed monocytes are truly hyper-responsive to chemokines. They migrate much faster and in much greater numbers into these tissues, and I'll provide you some evidence. Okay, and so of course what happens is you get too many monocytes, and as I'll show, hopefully convince you as well, dysfunctional monocytes that form and transform and differentiate into dysfunctional macrophages. Okay, and so why is this important? Well, very simple, because if you want to target macrophages in tissues, and some of you in the audience are doing that genetically and pharmacologically, that's actually quite difficult, because you're going to have to target the right ones, right? You want to target the inflammatory ones, but not the resolving ones. You have to get into the tissue, and you have to penetrate the tissue, and you've got to not kill the other tissues. If you could target the blood monocyte and prevent it from getting dysfunctional, logically, this component of the inflammatory process could be knocked out. And I can tell you, I won't show you any data. We actually are studying some compounds, and Nancy, my graduate student, yesterday presented on those. There are compounds, natural compounds, that actually prevent exactly this process. And they are surprisingly good at preventing atherogenesis and obesity. Okay? So that's the idea. And so I'll just very quickly go through the evidence that this actually occurs. So to prove that there is such a thing as priming, we use 3 mouse models of metabolic stress, an atherosclerosis model, an LDLR receptor deficient mouse on a low-fat maintenance diet, so with very little metabolic stress. If you feed these mice a high-fat diet, they develop dyslipidemia and very, very mild hyperglycemia. And we used the same model and made them die hyperglycemic by injecting multiple doses of streptozotocin. So if you will, a dose response of metabolic stress in vivo. Okay? That's how chemists talk. I'm sorry, I'm a trained chemist. And so what do you find? The question number 1 was, for us, because we were interested in oxidative stress and redox signaling, does it affect thiol oxidation? And to measure that, we took peritoneal macrophages from all these mice and measured the ratio of reduced to glutathione to glutathione disulfide. And I'll show you that if you use glutathione either as an electron donor or as an antioxidant, it will generate glutathione disulfide as a deoxidation product. So that ratio tells you something about how strong the oxidative stress in the system is. And you can see with increasing metabolic stress, you see a decrease in this ratio. So there clearly is thiol oxidation. You see a corresponding increase in lesion formation, atherosclerotic lesion formation. This is measured across the entire aorta of the mouse. Here, low, medium, high. These are the diabetic ones. And now the big question was, does it affect monocyte recruitment? And the short answer is yes. The way we measure this is obviously you can't count macrophages in the vessel because the vessel injury would obviously be affected by the metabolic environment. So what we did is we implanted Matrigel plugs that were loaded with either MCP1 or vehicle. So you have a well-defined chemoattractant gradient. And if the monocytes would not become dysfunctional, you would expect the same amount of monocytes in every one of these Matrigel plugs. But if the monocytes become hyperreactive, you should see within the same period more macrophages recruited in the plugs. And that's exactly what you see here. On the left side is the vehicle control. Here on the right side is MCP1 in the healthy mouse. You make them dyslipidemic. You get about 3 times more macrophages in these Matrigel plugs within 3 days. And if you make them diabetic, actually compared to healthy ones, you get 10 times more macrophages into these Matrigel plugs. And I guarantee you that cannot possibly be a good idea, even if these are healthy macrophages. Okay? And if you can actually use this to calculate the redox potential within cells, for those that are interested in redox chemistry. And the predictor of that redox potential, that oxidative stress, if you will, calculated based on this ratio, glutathione over glutathione disulfide, has a pretty good, it was a better predictor of atherosclerosis actually than cholesterol levels were, okay? Because actually these mice and these had the same cholesterol levels. So it suggested that yes, certainly there is oxidative stress, and it suggested that maybe this was the cause of that, but that of course was conjecture, and as such at this point was just purely correlative. So the question then became, how does metabolic stress actually prime monocyte? How does that work? Is it mediated by thiol oxidation? I can tell you right now, I wouldn't be standing here if it weren't. But more importantly, is monocyte priming sufficient to drive a relevant process, a pathological process, and in this case, atherogenesis? And then, of course, the ultimate question is, well, if it is, then can we prevent it? So now for the 3.5 minutes of thiol redox signaling in chemistry. So this is glutathione, and glutathione is made from cysteine. So glutathione is purely made intracellularly and cannot be taken up. Okay, so the cells control glutathione, and which is actually at millimolar levels in leukocytes, and low, high micromolar levels in other cells. It's your most abundant antioxidant, if you will, in a cell, particularly in cell culture, because your vitamin C and vitamin E have practically, particularly vitamin C is gone. This is the rate-limiting step, the attachment of a gamma-glutamyl, the glutamyl moiety to the cysteine, and then you add glycine, and then you have this very small tripeptide that is actually, people refer to it as a radical scavenger. It's a lousy radical scavenger. It's actually rather unreactive. It has to be deprotonated to react. But the reason it's a very good antioxidant is because it's an electron donor for a lot of enzymes, primarily glutathione peroxidases that can reduce anything from peroxide, peroxynitrite, and lipid peroxidases into the less toxic compounds. It is a substrate, or it is important for regenerating glutaredoxin, and we'll get back to that later. That is the enzyme that is actually responsible for recycling your dietary antioxidants, here, ascorbic acid. So the 2-electron oxidation product of the ascorbic acid or vitamin C back into vitamin C. And of course, vitamin C is used by the body and the cells to regenerate oxidized vitamin E, your primary radical scavenger in the lipid phase. Okay, but what glutaredoxin also does is catalyze a reaction that is called deglutathiolation. So and I'll talk about it in just for a few more minutes, but fundamentally, what happens is that reactive thiols, and very selective, only reactive thiols, usually the most reactive ones, and we can talk about why a little later, become oxidized, and glutathione becomes attached to these thiols. And this process is called glutathiolation. In most cases, this inactivates the enzymes or the proteins, or it causes them to mistarget. And we actually discovered at least 2 proteins that actually get degraded after they get glutathiolated. Glutaredoxin takes this back off. And you can already see if this happens fast enough, theoretically, this could be an activation-inactivation cycle, and that's exactly what it is. So I'll show you some examples of it. Okay? Now, the enzyme that then regenerates the glutathione disulfide, and that shouldn't be called oxidized glutathione because that's something else. This is glutathione disulfide. Back into its reduced form is called glutathione reductase. And the reason we know this cycle is really important in macrophages is because if you overexpress either the cytosolic version or the mitochondrial version in macrophages in an atherosclerosis model, that enzyme alone is sufficient to reduce lesion formation by over 30% over a 12-week period. So we know that this system is important. Okay? We just never really understood why. All right, so glutathiolation. So the concept is as follows. There are many papers out there. You can go read them. Glutathiolation has long been proposed to be a signaling mechanism, but it's very hard to prove it actually is, for various reasons, and I'll give you a reference that you can read up on. In part because it's very hard to detect these single thiols and their glutathiolation status. This system is fraught with artifacts. If you lyse cells, a lot of these thiols get automatically glutathiolated. So a lot of the data out there on glutathiolated proteins are actually complete artifacts because they didn't pay attention to it. But nevertheless, there are some very good examples of this actually being controlled in a redox-sensitive manner. The other problem was people never understood and tried very hard to find a thiol transferase that's actually responsible for attaching that glutathione to that deprotonated thiol, okay? That thiol anion. That thiol anion is not super reactive, okay? And it takes a lot of glutathione to attach it on its own. And in fact, it takes glutathione disulfide to do it non-enzymatically. So this has been a long mystery, and that's why people never believe this is real. But what I'm going to propose to you is that what really happens is we think that the step in between is actually an oxidation step that's catalyzed by hydrogen peroxide. If you oxidize this thiol to a sulfinic acid, this is very, very reactive. We'll immediately grab the first thiol in its vicinity. The most abundant one in the cell, as I told you, is glutathione. So this is one way to generate glutathiolated proteins. We always knew that glutaredoxin was an enzyme that's is specialized on one single reaction, and that is deglutathione and taking off the glutathione and releasing it as glutathione disulfide. Okay? And you can see here, if you will, the corresponding, or the analogy would be, here's your kinase activity, this is your phosphatase activity. Okay? And in most cases, like I said, this is your active protein, this is the inactive protein, with some exceptions. HIV kinase, for example, is one exception that gets activated as a glutathiolated one. Tissue factor 2 requires glutathiolation to be active, so there are a couple of abnormalities. This is the review I can highly recommend. John Meehl is an absolute expert in glutathiolation and glutaredoxins. It was published in ARS in 2008. If you're interested in this, read John Meehl's papers. And in fact, I'll get to that later. All right, so what I'm going to propose to you that in monocytes, we have this kind of process. We think there is an pH oxidase, maybe likely, at least in human cells, it's NOX4. Whether that's the case in mice is still controversial. That generates hydrogen peroxide, and again, you have the oxidation of the thiol, the attachment of glutathione, and glutaredoxin that turns it off. Okay, so we'll show you some examples of this. But, and I'll show you those data a little bit later as well. What I'm going to propose to you is that instead of a single stimulus oxidizing a single protein, if you expose cells to chronic metabolic stress, this system will modify somewhere around 700 proteins. At least that's as many we pulled out of the redox proteomics approach that we had. And I'll show you a little bit how they're distributed. Okay, so that was sort of the brief introduction. I, again, hope to have convinced you that metabolic stress actually does induce thiol oxidation and that it's important. The thiol oxidation increases macrophage recruitment, and we'll talk. And it appears to accelerate atherosclerosis. We'll provide you, or I'll provide you with some more evidence on that. And that thiol oxidative stress is enough and to sensitize monocytes to chemotactic signals, and that leads to what we call monocyte priming or monocyte dysfunction. So how does this work? Again, a lot of this is published, so I'll go through this pretty quickly. I'll just show you the in vitro model that we're using, and that would be this. These are a monocytic cell line, a human monocytic cell line, exposed to a chemoattractant in a chemotaxis chamber. Here is, if you will, normal migration of the response into MCP1, the chemoattractant. This is always the same amount of chemoattractant. If you pre-treat these human monocytes with LDL for 24 hours, human LDL, you double the response. If you treat them with high glucose, this was a little over 20 millimolar, you get an even bigger response. And if you add them together, you get a very robust and reproducible response. That's usually 2 to 3-fold higher than the baseline response. Okay, and so that's sort of our in vitro. This is our metabolic stress, if you will, in vitro. You can repick the hydrogen peroxide concentration that had this. So hydrogen peroxide accelerates chemotaxis. That was actually known. But we picked a concentration that had the same acceleration effect, if you will, as the metabolic stress. And if you measure, sort of estimate intracellular hydrogen peroxide levels, you can see that actually our metabolic stress conditions makes more intracellular hydrogen peroxide signal than the extracellular, the exogenous added hydrogen peroxide. And we can discuss what it is. Now, the easy explanation would have been you just change the receptor for MCP1. That wasn't the case. We've proven that. But more importantly, it doesn't matter if you use MCP1 or PDGF or RANTES. This priming effect seems to be universal. So it doesn't seem to affect a single protein. It doesn't seem to affect a signaling pathway. There's got to be something a little more global. Okay? This was just the evidence that it might be NOX4, at least in human cells, which is an inducible enzyme, constitutively active. Here's your phagocytic enzyme. Doesn't change during stress conditions. And I'm not going to show you any data, but most importantly, if you overexpress NOX4 in monocytes, you actually mimic this priming effect, and you can see the accelerated chemotaxis and the increased hydrogen peroxide formation. If you knock down NOX4 in human monocytes, at least, you prevent priming and the accelerated activation response. So we think we have at least some evidence that in human cells, maybe NOX4 is the source. It's very hard to pin down because it's expressed at extraordinarily low levels. But we still think fundamentally the principle of generating hydrogen peroxide to oxidize the thiol as the kinase part of this arm and glutaredoxin take off glutathione in the, if you will, analogous to the phosphatase arm of this. So this is what we think happens. And of course, by changing enzymatic and protein function, somehow this leads to this priming and hyperreactive phenotype. Okay, so I'll just show you a couple of example references. We've shown that actin is a target for this. Actin turnover is actually increased due to increased glutathiolation. That's been known. We could demonstrate it in monocytes, and of course, increased actin turnover is very likely to contribute to accelerated migration because you generate more actin, which can then be transported to the leading edge of the cell. So theoretically, the machinery you need to crawl is activated. I'll show you a little bit about MKP1 in a minute. And 1433-zeta is a binding protein that actually sequesters cofilin. And cofilin is involved in actin polymerization. And that too, 1433-zeta is, it actually does, sorry, it doesn't sequester cofilin. It sequesters slingshot phosphatase, which controls cofilin activation. But this enzyme, this protein too, is regulated by glutathiolation, is hyperglutathiolated in response to metabolic stress and degraded, releasing the phosphatase, which then increases actin turnover again. So these are three that we validated at a molecular level, knocking out the cysteine and showing that if you eliminate the cysteine, you don't get glutathiolation and the activity goes away, blah, blah, blah. But I'll show you this. Here, just to show you that this priming effect is not limited to an atherosclerosis mouse model. This is a C57 mouse that was fed a 60% high-fat diet, so a diet-induced obesity model. And again, these are Matrigel plugs, and you can see here are the plugs without MCP1, the plugs with MCP1, and this is the difference you get after, I believe, 10 weeks on a high-fat diet. And so you do get 3 times as many monocytes, macrophage recruited. In there, the macrophages we took out from these macrophages seem to have, and I say seem because we still need to confirm that again, an induced NOX4 expression. The reason I said seem is because unstimulated murine macrophages don't express NOX4, at least not at the mRNA level. We're not quite sure how that is, but we can detect it in activating cells. If you take the peritoneal cells, again, you can measure glutathiolation as a global protein amount of glutathione attached to protein. You can see glutathiolation occurs. And if you plot for each mouse the amount of glutathiolation or the amount of glutathione bound to protein versus macrophages recruitment, you get a pretty good correlation, suggesting there is a direct link between thiol-oxidative stress and this accelerated recruitment of macrophages into these plugs. Okay?
Reto, how do you measure HPLC?
So what we did is we did just a global IP of all proteins with TCA, washed it, then reduced the proteins with DTT and just measured the glutathione that came off the proteins. So very crude, but it was a simple way to get a global number of how much there is. Actually, by doing that, we realized that there was a substantial amount of protein that have glutathione bound in resting cells. So about 5% of all glutathione is already bound to proteins in resting cells. So that was interesting because we always had the concept that the only way you get glutathiolation is under conditions of oxidative stress. So clearly that wasn't correct.
Once you pull it down, you do mass spec or?
Well, we haven't done that at the time. Oh. I'll show you. We did mass spec in a very different way.
Oh, I see.
Because you don't get all the proteins that way, and you can create some artifacts. So, all right. So I want to show you one example, and it's probably a key player in monocyte function is this mitogen-activated protein kinase phosphatase 1. Okay, this is the phosphatase that inactivates ERK, so dephosphorylates ERK and dephosphorylates P38. It can do junk, but at a much lower activity, and it turns out this is absolutely critical for monocytes, and I'll show you that in a minute. And the question really was, what happens if this protein gets glutathiolated? Does it play a role in priming? And more importantly, if monocytes are primed, would it have long-term effects on macrophages that are derived from these proteins? So the reason this is an important protein is because adhesion in monocytes is controlled by ERK primarily, and chemotaxis, migration, is primarily controlled by P38. And in THP1 monocytic cell lines, you can separate it out completely. In human monocytes, there's a little bit of overlap. The activation of these pathways are counter-regulated by MKP1 primarily. And so the question was, well, if you inactivate MKP1 by glutathiolation of this reactive active center cysteine, then we should see a hyperactivation of ERK and more adhesion and a hyperactivation of P38 and more chemotaxis. And together, this should result in more macrophage recruitment, right? And if that is true and it is of any pathophysiological relevance, it should drive disease. And so I'm just going to show you a few data. Again, this was published a few years back. So the first question was, is MKP1 actually glutathiolated? And because that was really sort of the key piece of evidence for the paper. And when we first looked at it, we IP'd MKP1, and there was no glutathione, no difference. Okay, and so that was very disturbing. And so again, this was in vitro, exposed to LDL high glucose as metabolic stress. And then I realized there was an old paper in the literature, about 20 years old, that showed that crystalline is a protein when glutathiolated, and I didn't even know what that protein did. But if it gets glutathiolated, it gets degraded. And so we tried that. And if you add MG132 to this whole mixture and prevent the degradation of the protein, you can see that the amount of glutathione attached to the phosphatase is enormous. Okay. And if you mutate out that specific cysteine, there's no glutathiolation whatsoever. So it's only one cysteine out of 7 that actually gets glutathiolated, very specifically the active center cysteine. And of course, we couldn't work with this protein because that's dead. So there is no activity. But it proved the principle that the glutathiolation is a highly specific process, okay, site-specific.
So Reto, how do you measure the glutathiolation level?
In this case, we IP'd the protein and use an anti-glutathiolation antibody. And if you have enough protein, that antibody is sensitive enough to pick it up. It's very unspecific, so you shouldn't use it in whole cell lysates or whatnot, and you can't really IP with it. But if you have a good antibody to IP your protein of choice, you can use that, as a detection model. It's very, very sensitive. And so for this paper, we did a bone marrow transplantation experiment and actually transplanted atherosclerosis susceptible mice either with wild-type bone marrow or with MKP1 deficient bone marrow. Did the same Matrigel assay to see whether priming occurs. And again, you see a very nice 3-fold increase, roughly, whatever that is, maybe nearly 4-fold, increase in macrophage recruitment to the identical gradient of MCP1. Okay? And so, then in the same mice, we took out the aortas, and you can see a very nice acceleration of atherogenesis. Okay? Again, it's not causal, but it's a good, at least we're an association by guilt, at least, right? But we're narrowing it down. Okay, so this was sort of the concept back, 3 years ago, that the adhesion and chemotaxis of these monocytes that get recruited are really controlled by the brake that we now call MKP1. And the activity of this brake seems to be under the control of a redox-sensitive cysteine, right? And if you eliminate it, like the brake, and you don't have the foot on the gas pedal, if you don't have MCP1, nothing happens to the cell. They migrate just fine. They look the exact same way. But once you put the foot on the gas pedal, i.e. you add your chemoattractant, and there's no brake there, that's when they start running toward their goal, and you see this hyper-recruitment. And in the case of, like I said, metabolic stress, we have a source of reactive oxygen species, presumably NOx4, and then that leads to the degradation of MKP1, and effectively there is no more brake left at all. It's not just inactive, it's actually being degraded. Okay? And we think that that is a key process in increased accumulation of macrophages, and you can imagine, we'll get to that a little later, this is probably not just true for large vessels, this could be true for the adipose tissue, for example, and I'll show you an example of obesity a little bit later on. So the question now was, as I told you, the half-life of a monocyte is 2 weeks. Okay? So who cares if they're dysfunctional? The half-life of the inflammatory monocytes is 1 to 2 days. We couldn't care less if they're dysfunctional. Okay? But if the priming effect would be something that the cell remembers, and remembers past its differentiation state into a macrophage, then you potentially have a problem. Because those macrophages can now live for months, and in fact, in an atherosclerotic vessel, they can live for years. Okay? So that is your real problem. So that was an important question. The only problem was, how do you address that? And so our way to look at this was the following. We knew that in diabetic mice, we actually isolated blood monocytes from individual diabetic mice and measured MKP1 activity. There was way too little protein to do anything with them, but you could measure MKP1 activity. And it turns out that in these diabetic DB mice, you get an over 50% reduction in the activity of the protein. Okay? So this is probably actually a very nice marker of early metabolic stress. Okay? And so the question then was, well, if we use these same MKP1 knockouts and follow them into the site of injury, in this case into the atherosclerotic vessel, would these MKP1 deficient macrophages also show dysfunction with regard to parameters that are important for atherosclerotic lesion formation? And what we looked at is autophagy and apoptosis and macrophage polarization. So we knew, of course, I showed you already, that they accelerate atherosclerosis, and any defect in these parameters would probably drive it. So, and I'm just going to show you some selected. This is all unpublished. Hopefully, it's going to go out to Cell Metabolism in a couple of weeks, hopefully. Here we look at autophagy as a marker. We use P62. So the more P62 you find, the worse the autophagic flux in the cells. CD68 is our macrophage marker, and so we looked at overlap between green and red, and so that would be yellow. And so these are mice that received wild-type bone marrow. Here is a section through an aortic root. Here you see the cusps and the valves, and you can clearly see there's a lot more of P62 expressed and detected in these hearts or these aortic roots from mice that actually had macrophages that were MKP1 deficient. Okay. Now remember, this in the diabetic mice I showed you, it was only 55% reduction. This is 100% deficiency. Okay? But nevertheless, it points out that this single protein clearly has dramatic effects on macrophages within the vessel wall, and half of that is already missing under conditions of metabolic stress. This is just a quantitation of the data. I can tell you these MKP1 deficient macrophages are very susceptible. Go ahead.
Do all macrophages express CD68?
Yes.
I thought CD68 was more of a marker of expression of activation in macrophages.
No, actually the problem with CD68 is that a lot of other cells can express CD68, at least at the mRNA level. But macrophages express far more CD68 than other cells. So neutrophils have CD68 at low levels. Smooth muscle cells have CD68. No, it does change. That's correct. But it's still one of the more specific markers of pan markers of macrophages that you will want to look at. But you shouldn't look at it at the mRNA level. Then you get into trouble. So people normalize macrophage responses to CD68 mRNA levels. That's a very dangerous thing to do because we know that other cells, adipocytes, for example, express CD68. So normalizing in adipose tissue against CD68 is probably a very bad idea. You're going to have to use something else or fax them out. But very good question. These macrophages are also very, very sensitive to oxysterol-induced cell death. So alpha-ketocholesterol is one of your major oxysterols in any site of oxidative stress and inflammatory situations, atherosclerotic lesions being the prime ones. They die far more easily than wild-type cells, which is probably going to contribute to the acceleration. And the more interesting question was, well, would it affect macrophage polarization? Okay, and I don't have time to get in all the details, but the principle is you can have resting macrophages, which we'll call M0 for the time being. This is the same principle, oversimplified, that you have in T cells. You can polarize them into Th1 or Th2 cells. In macrophages, that's where they stole this nomenclature, M1 and M2. There's a lot of controversy. This is probably a spectrum of polarization states. It is not just a spectrum of polarization state. It is highly microenvironment dependent, so they will look different in different tissues. But it's useful if you take it for what it is, and that is the argument that this is a certain program where you expect a certain number of changes. And what you'll see here is we picked, I think, 20 markers of M1. So one thing you should never do, if you want to look at polarization, pick your 3 favorite genes, see whether they change, and claim that you looked at polarization. Okay? Because depending on where you pull those macrophages out and the way you treated them, chances are 2 of them change anyway. Okay? So what we did is we designed fluidine chips, and we routinely measure polarization by looking at this whole slew of M1 markers and this whole slew of M2 markers. And depending on the conditions, not all of these are going to go in the same direction, but the majority should. Okay? So let me explain. So these are M0, so these are resting cells from either wild-type mice, and we normalize the signal from these genes all to 1. Okay? These are the MKP1 knockout mice, and you can already see that anything in yellow, here's the scale. Yellow, 1 to 10-fold increase, orange, 10 to 100, and red is over 100-fold increase, and the same in the other direction. So green is suppressed, red and yellow is increased. And you can already see that the knockout mice have slightly different expression levels for these genes across the board. Okay? So clearly, MKP1 deficiency already changes something. Okay? Now, if you polarize them into an M1 phenotype, when we use TNF-alpha plus interferon gamma, which is pretty well accepted, actually a good model to use, here's the wild type. So you see all these M1 genes go up to a certain extent, and they're ordered by the degree of increase in expression. So they're the same genes as here, they're just in a different order. But what you can see in the knockout, this increase in M1 actually is more pronounced for just about all the genes. Okay? So MKP1 deficiency sort of enhances the look of an M1. Okay? And then if you treat them, instead of with TNF-alpha and interferon gamma, if you treat them with IL-4, here we go, then you see very little happens for these M1 markers. Right? They stay pretty close. But if you look at the M2 markers now, you see that a lot of these changes are actually suppressed now. Okay? And if you assume, and we're pretty sure that the M2 is not an anti-inflammatory macrophage, that's actually probably a misnomer. They do release inflammatory mediators, but these mediators are designed to resolve inflammation. Okay? That's probably what the main role of these M2 cells are. So we have the pro-inflammatory and the inflammation-resolving phenotype. And so you can see if this one, M1s are more pronounced and M2 suppressed, you're shifting a balance. Okay? And if this were true for primed cells, well, yeah, then it is important what the monocytes see in the bloodstream because they're going to remember by the time they get into the tissue. Okay? And so, like I said, MKP1...
Yeah. So that was said it's a microarray?
Yeah, it's a fluidine. It's a specialized PCR microarray that we designed to measure these things routinely. It's a nice tool that we thank Tim Huang for the facility. But it's an easy way to monitor a lot of genes at the same time. And you can design it yourself. So we made these, for example, for metabolic genes, and so it's a really nice tool to screen a limited set of genes and with small samples of RNA. These guys can do it with single cells, so we're happy if he can do our 50,000 that we isolate from our orders. Again, so just to remind you, again, in diabetic conditions, you don't have 100% deficiency. Okay? You have 55. But very likely, this deficiency in the diabetic monocyte is going to impact autophagy, apoptosis, macrophage polarization in the diabetic vessel as well. And it's probably going to, very likely, going to contribute to accelerated atherosclerosis associated, for example, with diabetes. Okay? So the question really is there metabolic memory? And so we did the same experiment now. Instead of using knockout mice, we took peritoneal macrophages and primed them in vitro for 24 hours. Okay? And so this was a little bit of a long shot, but as you can tell, it actually worked. So here are, again, the controls. Everything normalized to 1. LDL high glucose, 24 hours. You can see that already changes the base, if you will, the base program. Not very much, very lightly, a little up and down, but it's not the same thing. And it's hard to see here, but this number is 150. Okay, if in the primed cell, it goes to 190. This was 145, goes up to 420-fold increase. And so, again, across the board, we see some, not all genes, but some genes change quite dramatically after priming. Okay? And the only thing we've done is expose them to metabolic stress, and yet the gene expression profile is already altered. And the same is true for the M2. The M2s, you can see this is here 6,700, gets cut down to 4,000, 105 gets down to 32, 24 is only 12-fold. You can see a dampening of the M2 response, very similar to what we saw in the MKP1. Okay? So you can mimic this effect of MKP1 deficiency by simply exposing tissue macrophages to metabolic stress. That doesn't prove anything, but it suggests that the cell might be able to remember. So the question really is, how do monocytes maintain this memory? Okay? How is that signal sensed, and how is it translated into changes in transcription of so many things? And so what I'm going to propose to you is that what metabolic stress does, it actually reprograms the entire proteome of the cell. And it does that via these glutathiolation modifications. And in order to prove that, we designed a redox proteomics approach. And with that approach, we tested again and compared THP1. So I'll just show you very selective data. This just came back, so this is going to be published hopefully very soon. We're publishing actually editing a forum for ARS on macrophages. Some very interesting reviews, one on epigenetics in macrophages, one on tumor macrophages, one on redox signaling in macrophages, and one on, I forget, I'm blanking. I'll tell you later. And this paper will be in there as well. But the interesting thing here is, these are THP1s, okay? So there were roughly 680 proteins that were identified at least once. We only focused on macrophages that we found in all 3 sets of experiments, and those were about 133. Interestingly, only 6% of those are de novo glutathiolated. They start out not glutathiolated, and glutathione is attached, okay? That's a very low number. So most of them are already modified. It's just that the modification level changes. And interestingly enough, a lot of them get deglutathiolated under conditions of metabolic stress. They're all over the cell. I can show you the list, but about 3/4 of them are cytosolic. 14% of them are nuclear. Okay, so here is where it gets interesting with regard to reprogramming of the cell. But you can imagine if you change the expression level, the activity, and or the localization of 680 proteins, something is bound to happen to that macrophage. When we did the same experiment and compared metabolic stress with hydrogen peroxide, the same hydrogen peroxide condition I showed you that had the exact same chemotaxis response early on. Okay, it's about, these are tumor cells, so you can throw a ton of hydrogen peroxide on them, about 3 millimolar for an hour. And what you see is that surprisingly, really, if you think about it, 94 out of these 133 proteins were identical, and it didn't matter whether you added LDL high glucose or hydrogen peroxide. Now, hydrogen peroxide is probably going to oxidize the thiol directly, okay? But there were some differences. There were a few more that were oxidized with hydrogen peroxide, suggesting, yeah, throwing extracellular hydrogen peroxide in the cell is probably not the best model of intracellular oxidative stress, but nevertheless, it modeled quite nicely. Now, what was far more interesting, and the question for us was, is this even relevant to a physiological, pathophysiological condition? And so what we did is we went back and used macrophages from mice that were fed a high-fat diet. The problem is, these are mass spec experiments. You need a ton of mice. We had to pool virtually 10 mice in each group to just have one sample, okay? But it was worth the while. So these are all maintenance-fed diet, fed LDL receptor mice. These are high-fat diet-fed mice. And you can see 46 of the 74 proteins were in common. 11 were unique to the maintenance diet, and 17 showed up only after high-fat diet. Now, I can tell you that many of these actually changed levels. Okay, there were many more differences. I'm just showing you the identity in which one showed up. What was far more interesting is if we looked at male and female mice. Again, we isolated these macrophages, cultured them, then analyzed them. And if you compare males to females, only about 53% of the proteins that were glutathiolated were actually glutathiolated in both male and female mice. Okay? And the other important thing is females have many more proteins that are glutathiolated than males. Okay? 74 versus 120. And in this particular mouse model, you should also know that females are far more susceptible to develop atherosclerotic lesions than males. And, I will show you later, they're also more susceptible to get fat. Okay? So maybe this is a dimorphism, sexual dimorphism, that might be relevant to disease progression. Okay? And we're looking into this. There are actually some very interesting differences in proteins between males and females that we can maybe talk about later. So, I'll just leave that here. The question now is this kind of change of reprogramming maybe the step before epigenetic changes occur? And indeed, we know, for example, histone 3 gets glutathiolated in response to high-fat diet. We know that some of the epigenetic writers and erasers are getting glutathiolated. Some very interesting things happen in these cells, and we're still looking at it. The problem is we're actually not sensitive enough with the mass spec technique. We probably just skimmed the surface. And we know that because, for example, MKP1 didn't even show up in the screen. Okay? So we just found the most abundant ones, and many of those were metabolic enzymes. But they can explain why metabolism changes in these macrophages. Okay? And very interesting example is actually pyruvate kinase. Pyruvate kinase behaves differently between males and female macrophages. All right, so before we go to the very last piece of the talk, and I'll rush through, so I hope I've convinced you that protein glutathiolation, deglutathiolation is indeed a signaling paradigm that works, and an important one, a physiologically relevant one. If you activate this paradigm on globo with metabolic stress instead of a specific stimulus, you then actually end up reprogramming the monocyte proteome, okay? By altering activity, expression levels, and like I said, targeting in the various compartments. And I hope to convince you that it's probably a reasonable assumption that macrophages derived from metabolically primed or dysfunctional monocytes will show dysfunction after post-differentiation, okay? And we're working on that. We're trying to show you some more concrete evidence. So, very quickly, final thing, an interesting story that developed a little over a year ago gets at the question, does obesity and cardiovascular disease have something in common? That sounds like a really stupid question, but it's actually not quite as stupid as it sounds. If you want to know whether there is a correlation, then you can look at these maps, right? I find them interesting. So here, this is the prevalence of obesity. It's 2014. So here's our obesity hotbed, okay? Now, unfortunately, I don't know why these statisticians can't agree on a single graph. Every single map you find for different diseases has different colors and different sizes. But this is cardiovascular disease, and here you go. Right there, smack in the middle. At the same hotbed, you find a lot of cardiovascular disease. Again, it's guilt by association, but nevertheless, there could be something in common. And what I'm going to propose to you is that the commonality between obesity and cardiovascular disease is, in fact, the macrophage. And why should that be? So before I forget, so this is actually something my graduate student has been driving for the last year. He's done a great job. We probably would have overlooked this and postponed this experiment forever if he wouldn't have made a very interesting observation. He is a truly gifted grad student with very gifted hands. If I can just get him to sit down and write, I would be a very happy camper. Anyway, so why should there be a commonality? Well, because even though this slide is made for an atherosclerotic or for a vessel, the process of recruiting monocytes into tissues, them differentiating into macrophages and then polarizing is not unique to the atherosclerotic vessel. The same thing happens in the adipocyte, okay? Except that the adipose tissue is already loaded with M2 macrophages. The monocytes that come into the adipose tissue become M1s, and that's how you now shift the balance between the two. Okay, so here's what he found. I told him to go down and get me a couple of glutaredox knockout mice because we were going to do some ex vivo experiments to see whether the other side of the equation, not the oxidative stress side, but the, if you will, the phosphatase side of our signaling paradigm, if you muck with that, whether you could have similar effects as increasing oxidative stress. So he comes back and said, "Your mice are all screwed up, man." And I said, "What do you mean?" He said, "They look like this." And so what happened is, we delayed this experiment so long that these mice were over a year old. Okay, and that happened to be the only ones we had. We were running low on money. I told them to kill the other ones off and just keep a couple breeders. And so these, as it turns out, this is a 1-year-old glutaredoxin-1 knockout mice. And for those, this may not be as surprising, but for those of us in the field, glutaredoxin knockout mice have been used for a lot of things. And they were phenotypically a failure. They didn't show any real significant phenotype. They were boring. And in part probably because glutaredoxin there are isoforms of glutaredoxin that are expressed both in the cytosol as well as the nucleus and whatnot, and they probably compensated. But this was really interesting. So the question now was, well, this is a whole body knockout. How much of this is actually due to glutaredoxin deficiency in macrophages? Could we create an obese phenotype by simply knocking out glutaredoxin in macrophages? And, oh, these are just the growth curves. So one of the interesting things here is, again, In red are the females, okay? So these are wild type. And look at these transgenic mice, or knockout mice, if you will. So the females are much more susceptible to becoming fat than the males, okay? But the males will do it at the same time. And the reason you don't see it is because this is months. Unless you wait at least 6 months, you wouldn't even detect any change. And by that time, most groups had actually knocked out their mice. Unfortunately, 2 other groups in the US have had a similar smart graduate student, apparently, as I did. So they're on this as well and studying this, so we're going to rush. But again, as I told you, what we're looking at now is instead of increasing stress on this end, what happens if you eliminate the relief reaction, the phosphatase reaction, okay? So again, the question was, what happens if you knock out glutaredoxin macrophages? Does that in itself, is that enough to drive a metabolic phenotype, and is that enough to drive obesity? And the answer is yes, okay? So the only thing different between these cells is really the expression of glutaredoxin in the bone marrow, and primarily in this case, macrophages. And you can see these are females. And again, here after about 6-7 weeks, the curves start to segregate. Okay.
Radiation primarily?
Yes. Yeah. So these are irradiated, bone marrow transplanted. And so we'll have to do this again in a more, in the knockout and putting them back with overexpressing transgenic or GRX1. So we're preparing all that. We have the vectors, we just haven't done the experiment. And as you can see, sort of after about week 6, 7, 8, here the weight starts segregating, right? And this is where we did the takedown. We knew that we wanted an early point where the weights were barely different, so we took them down. We measured atherosclerosis, both here after 6 weeks. We knew there wasn't much. And then after 20 weeks, where you had nice segregation of the weights. And now if you look at the glucose levels, you can see about the same time, so it's week 6, 7, 8 to 10, is where the glucose levels start segregating out, so they become hyperglycemic. And again, the only thing that's different is that these macrophages don't express GRX1. Nothing else changes, okay? So here's where things start getting interesting. They become hyperglycemic, probably due to the fact that they're becoming fat, okay? So chances are this is the result of the obesity phenotype, okay? So the big question now is, are the macrophage dysfunctional before this happens and driving this? Or are the macrophages just becoming dysfunctional because they become obese, because these mice become hyperglycemic? And so at week 6, where we see no real difference in glucose, no real difference in obesity, we did our Matrigel assay to see, do we see a difference in macrophage recruitment? Oh, we measured atherosclerosis, so these are the entire aortas. You can stain them with Reto. You measure lesion size, and you can see by eye that there's quite a bit of difference. This is the quantitation. Again, no atherosclerosis whatsoever, really, at week 6. And as expected, nice lesions at week 20, and quite a bit more in the GRX1 deficient mice. And again, this is really only macrophage driven, right? So they become obese. They develop atherosclerotic lesions quicker. They become hyperglycemic faster, probably because they become obese. But are the monocytes dysfunctional? And they are. So this is at week 6. Okay. Nothing happened yet. No lesions, no obesity, no hyperglycemia. But these monocytes are already much more primed, much more hypersensitive to a chemoattractant stimulus. And so we think that this whole concept that the redox balance in the macrophage is really not just controlling individual proteins, but the entire proteome of the macrophage, and mainly probably metabolic pathways. That's actually what we're looking into right now. That by changing the metabolic enzymes, they change flux of metabolic substrates, which then directly can lead to oxidative stress, but also changes in gene expression, protein translation, etc. Okay. So this is all still preliminary. So again, this is how we think things happen. In response to metabolic stress, you see thiol oxidation, protein glutathiolation. That then leads to the reprogramming of the proteome. That probably happens very quickly, within days. Then we can measure monocyte dysfunction, again, within days. We're looking into the concept that we see epigenetic changes. We actually have a grant funded by the Southwest Research Foundation to study this in baboons. So we're actually feeding these baboons 2 different diets and going to chase down and see whether the epigenetic landscape of the macrophages from these baboons changes, because within weeks, long before they have vascular changes or anything else. So that'll hopefully address this particular question. We think that that then leads to the reprogramming of the macrophage. which is then responsible for these altered responses within sites of inflammation. We have a more pronounced inflammatory phenotype, a suppressed inflammation resolving phenotype. And for those of you who are familiar with atherosclerosis, the current theory of why humans really develop atherosclerosis is not because they become more inflammatory, but mainly because the human vessel is incapable of mounting a good resolution of inflammation. The best they can do is a fibrotic response, stabilizing the plaque, but that's what you see. After a while, all you see is these fibrotic, stable plaques all over, and if you're unfortunate, some of them are not quite as stable, they can rupture and then you get a heart attack. So we think this is really, truly macrophage-driven, okay? At least in the early phases. And we think that this shift is what leads to chronic inflammation, be it in the adipose tissue, be it in the vessel. And in fact, these mice, these GRX1 bone marrow chimeras, develop massive steatosis. We find increased macrophage recruitment in the liver, lipid depositions in the liver, and we also find a very significant increase of macrophages as it looks right now in the muscle. So maybe the excess recruitment of macrophages into the muscle leads to insulin resistance there as well and drives this phenotype across the entire animal. Okay? So there may be more than just a connection between obesity and atherosclerosis. This may be connecting metabolic disorders in tissues across the phenotype, at least everywhere the macrophages go. And so I'll leave you with this. We think that monocyte priming and glutathiolation in response to metabolic stress is an explanation, a molecular explanation between an association we've long known between metabolic disorders, oxidative stress, I would call it thiol oxidative stress because that's what we can actually measure, cardiovascular disease, obesity, and maybe even other chronic inflammatory diseases. And so with that, let me just quickly, last 30 seconds, thank everybody. So the people involved here, a very talented postdoc now, a professor back in Korea, Hong-Soo Kim, he did all the MKP1 studies, all these papers. Nancy Nugent just defended her thesis proposal yesterday. She works on, as a graduate student, working actually on the prevention, dietary prevention strategies based on this principle of monocyte priming. She has an interesting new compound to work with. Kevin Downs, perseverance galore, an outstanding graduate student who did all the work on the GRX1 knockout, and other people in the lab. Sarah Ulovic was now a professor at UTSA. She did the early work. She did the redox proteomics work. Shifeng Li did the work on NOX4 and the early work on macrophage death. And our collaborators, first and foremost, John Meehl, who even put that bug in my head 15 years ago that redox signaling should be something really important and worth studying. And of course, thanking our funding agencies. And thank you for listening.